Hello there, invertebrate zoology class. I want to finish off the arthropods today by talking one more time about the crustaceans. um, and so we'll start with this sort of cartoon-like figure here of a typical crustacean. We can see here at the head end here in the first bracket, we have a number of segments, but they're hard to see, right? They're hard to tell how many segments there are. There's usually not any dotted lines here. So we've got perhaps a pair of compound eyes, but we have two pairs of antennae. That is a quintessential crustacean character. All crustaceans have two pairs of antennae. In addition, there are three pairs of mouth parts, a pair of mandibles, and two pairs of maxillae. And so it is therefore estimated that there are 1, 2, 3, 4, 5 segments on the head. Then we have the thorax region of the animal, which is oftentimes covered by the carapace or shield. It covers the head and thorax, right? In the thorax region, we have three pairs of legs that are oftentimes used as mouth parts. And those first three pairs of thoracic appendages here are called maxillipeds. So first, second, and third maxillipeds. Then we have a series of walking legs, oftentimes 1, 2, 3, 4, 5 pairs or 10 walking legs, such as in the decapods, the crabs. Finally, we have the abdomen here with a bunch of segments, excuse me, and we have abdominal appendages here, or swimmerets, and then last, a telson or tail with uropods around it. That's a typical crustacean cartoon, and this, of course, is more realistic. Let's go through this then briefly. Here's the carapace covering the head and the thorax, or the cephalothorax, right? Just one big shield, carapace. Again, we have a pair of stalked eyes, antennules, and antennae. Okay, the two pairs of antennae, again, defining characteristic of the crustaceans. We have mandibles and two pairs of maxillae. Those are all head appendages. We have a thorax with 8 segments, right? 5 pairs of walking legs, plus 3 pairs of maxillipeds. Okay, the walking legs are also known as periopods, and the abdominal appendages are also known as pleopods, right? So abdominal appendages, pleopods, thoracic appendages, periopods. Often, but not always, the first pair of walking legs has a claw or cheliped on it, like in a lobster. Okay, and at the tail end here, we've got the telson and the final pair of appendages, the uropods. Okay, so let's take a look at a crayfish then. Looking from above here, what we're seeing essentially is just the walking legs. All the mouth parts are underneath here, which I'll show you in just a minute. So here's the first pair of walking legs, second, third, fourth, and fifth, right? And the first pair have very large chelipeds or claws here. But if we flip the animal over, we can see that right here in the mouth region, there's a whole series of appendages. Really, the most easy ones to see are the outermost here, the third maxillipeds. But as I'll show you in a minute, if we begin to take those appendages off the mouth, and then the ones below them off, and so on, we're going to literally go through 3 pairs of maxillipeds, and then 2 pairs of maxillae, before we get to the hard, calcareous, grinding organs called the mandibles. Okay. Now, in this particular figure, we can also see, when comparing A with B, that ventrally here, underneath, in the male, the first pair of pleopods here in the abdomen are hardened tube-like structures called copulatory appendages here, which transfer sperm. There's 2 pairs here, 4 in total, that all come together to form a tube to transfer sperm over to the female. So the female just simply has swimmerets here, the entire abdomen, whereas the male has these long, hardened first set 2 pairs of copulatory appendages. All right, so what we're going to do next is we're going to begin to look at these appendages in the mouth area. Now, for each appendage, each one is biramous, okay, meaning there are 2 branches. There's an exopod, oftentimes shown in yellow, and in the next couple of figures here, an endopod, which is generally shown in purple. At the base of these legs here, the coxa, is oftentimes attached to the gill. So when you grab the leg by the base and pull, you oftentimes pull off the gill and, with luck, both parts of the two-part or biramous appendage. So here in this figure, the exopod is in yellow, the endopod is in purple. You can see, for example, the final pairs of appendages right next to the telson here, the uropods, have both endopod and exopod. Okay, but not all of these are biramous, to be fair. So if we pluck the appendages off, we have the antennules and the antenna, right? Again, crustaceans have two pairs of antennae. And then if we start from the outermost area in the mouth area of the crayfish, we'll pull off the third maxillipeds first, and then the second, and then the first, and then the maxillae, which are our head appendages, and then the first maxilla, so second and first maxillae, and then the mandibles, right? So the head appendages are the second and first maxilla, the mandibles, the antenna, and antennules. 1, 2, 3, 4, 5 pairs of appendages, 5 segments on the head. Okay, the thoracic appendages, again, we have the first walking leg, the caliped, the second, for some reason they don't have the third here, the fourth, right, and the fifth, they don't have that either, right? So, and then swimmerets here. On the female, all look feathery and are for helping with swimming. On the male, the first 2 pairs are hardened and form these tubes. Okay. Internally, if we take a look inside a crayfish, we can see its digestive tract quite clearly. The opening here, the mouth, right, next to the mandibles, right, enters a short esophagus into the stomach region here where food items are continued to be ground up and sorted and then shunted down a long intestine, and then feces, of course, are exiting out the anus. You can see in this sagittal section here that the heart here, which has openings or holes, ostia, the ostia allow the blood to be sucked into the heart, and then those valves close, the heart contracts and pumps the blood around the body. The space around the heart here, the pericardium, is strictly speaking the true coelomic cavity. We also have a very large digestive gland near the stomach area here. And if we take a look at the stomach a little bit more carefully, we can see that the stomach is really, there are 2 parts to it. There's here's the esophagus entering here, the food entering in these blue arrows. Here's the cardiac stomach, which literally has teeth in this gastric mill area for grinding up the food more. And then we have this pyloric stomach, the second region here, which has a series of filtering setae for filtering out particles, some of which are shunted to be stored in the cecum. Others, of course, are shunted down the long intestine. In terms of excretion, crustaceans have an antennal gland or a green gland. It's called green gland because of its color, and it's called an antennal gland because it empties here, right at the base of the antennae. In fact, when lobsters signal to another lobster that they're hiding in a den and this is my den, beat it, that signal takes a form of urine secreted out of the antennal glands, flowing out of the entrance to their den. So it's a chemical signal. Now, nitrogenous waste, primarily in crustaceans, are diffusing out of the gills. So this particular green gland or antennal gland is more often than not used for osmotic or ionic regulation. So crayfish, for example, it would be pumping out excess water, which is building up, of course, inside the animal's body because its body has more salts than the exterior fresh water. Life cycles vary, of course, but one characteristic is found in all of the crustaceans that are found in the aquatic realm, that is, and that is this nauplius larva. It's a characteristic larval form of crustaceans. So once the egg is fertilized, that embryo develops into a nauplius larva that swims around and feeds in the plankton. That nauplius might go on to form a protozoal larval stage, such as in these Gulf shrimp, and then a mysis stage, a post-larval stage, and finally an adult stage. So multiple stages, of course, each of which have multiple molts, okay? And that is a Gulf shrimp life cycle. In contrast, a crab life cycle, such as the Dungeness crab we find around here, here's the adult. The adults release sperm, male, or eggs, female. Fertilized eggs then develop into a zoea larva, shown here in the upper right figure here. Note the big spine on its helmet, primarily to keep it from being eaten by predators. And then that zoea will eventually molt and develop into a megalop form here, megalopy. And this megalops form here basically looks like an adult crab with this abdomen sticking out. And it is much heavier, tends to sink to the bottom, fold its abdomen underneath, and voila, we have the juvenile crab crawling around, developing into an adult. I want to finish off arthropods by talking very briefly about insects. And insects have mandibles just like crustaceans do. In fact, most recent evidence suggests that insects may actually be just specially derived crustaceans, believe it or not, evolved from within the crustacean group. Insects have a couple of common characteristics. They have a fusion of the second maxillae to form a lower lip called a labium. They also have a loss of all their abdominal appendages. Okay. Insects and many arthropods have a compound eye. And here's the compound eye of an insect. Imagine this big spherical eye, which is really made up of many, many little individual eyes, hence the term compound eye, right? And each individual element here is called an ommatidium and has a corneal lens, which focuses the light down through a crystalline cone area onto these individual cells here in the rhabdome. And these cells that detect the light are called retinular cells. Surrounding them are pigmented cells to prevent the light from entering into the next adjacent ommatidium. So a compound eye then is made up of many, many, many individual ommatidia, which are basically little eyes. If we take a cross-section through the ommatidium of a typical insect, you can see here in cross-section that something that's characteristic of all mandibulates, they have 8 retinular cells. These are the pink cells. If you count them here in this figure, there are 8 in cross-section. It's a very fine detail, but it may be important for linking them together as mandibulates. Okay? This type of compound eye is excellent at detecting motion, not as good at forming a 3D image compared to our camera eye. So an insect, for example, buzzing about you and you're trying to swat it, is very good at detecting the motion of that fly swatter and avoiding being killed, usually. Much better than we would if we were flying in that size through the air with our image-forming eye. Here's another image here of the individual ommatidium with the lens, crystalline cone, focusing light down through and onto these retinular cells, which in cross-section here you can count 1, 2, 3, 4, 5, 6, 7, 8 retinular cells. Okay, in terms of respiration, insects use trachea. What are trachea? Well, they're basically tubes here, the opening of which is called a spiracle, leading down through this tube or trachea down to very fine tracheoles that literally branch and allow oxygen to diffuse in through these tubes right directly to individual muscle cells or other types of cells. And again, these are found in insects and many spiders, and they are very highly branching within the animal's body, delivering oxygen directly to the cells. And their ability to carry oxygen in and finely branch sets an upper limit to the size of insects. Can't get larger because these trachea can't get more finely branched and literally supply oxygen to all parts of the body. In terms of excretion, insects use something called Malpighian tubules. And Malpighian tubules are really quite distinct from most excretory systems and invertebrates because they connect with the digestive tract. This is one place where confusion might actually lead to the right answer. Many early zoologist students are confused about excretion. They think excretion has something to do with formation of feces. Now, feces are formed by the digestive tract. Yes, they are wastes, left over, undigested portions, right, of your meals. But we're talking about creation of urine, right? Your urinary tract. So the equivalent of the urinary tract in insects is the Malpighian tubules. These Malpighian tubules soak in waste here in these arrows into these tubes, which literally, in this case, connect to the hindgut here near the rectum. And these wastes that are gathered by these Malpighian tubules then travels with the feces down through here. Water solutes are reclaimed, and literally the feces are coated with the uric acid crystalline wastes around the outside of them. Okay, so we have feces and excretory products all together coming out the anus. Here's another picture of this. If you imagine, here's the midgut and here's the hindgut. Here are these Malpighian tubules entering. If we take a look at each of these tubules, uric acid crystals are being formed within them. Sodium, potassium is pumped in along with water, sugars, and amino acids, which tend to diffuse in, right? And then as those products are traveling down through the hindgut with the feces which are forming here, water is reclaimed, sodium, potassium, sugars, amino acids are reclaimed, and out of the rectum comes these feces, right, poop, which is coated with uric acid crystals. It's a great system for preventing a lot of water loss. Okay, now the next thing I want to talk about very briefly is flight. And in order to understand flight, we again need to go back to the outer exoskeleton here, right, which is molted for the animal to grow, right? And think about these individual plates here, the tergum on the dorsal part of the animal and the sternum down below here, along with these pleuron here, or pleurites, okay? And there can be bending and movement of these plates in certain areas that are thinner, like this here, articular membrane. So, when we think about flight muscles of insect, flight muscles shown here either directly attached to the wings, in which case they're called direct flight muscles, okay, found in locusts and dragonflies, for example, or they indirectly attach. What do you mean? They indirectly attach here and here to the upper region of the exoskeleton, right, which would be the tergum, okay? So these muscles then are going to bend in the tergum. Imagine that outer convex tergum is going to bow in now. What's going to happen to the wings? They're going to fold upward right here, shown in figure B. So that's indirect flight, basically flight not by muscles moving the wings themselves, but by moving the actual exoskeletal regions here in areas that are meant to be bent. Note here, insects evolved flight long before birds, and that open niche, flight, air, right, allowed an incredible diversification of insects. Okay. In terms of evolution of these different groups of arthropods, I have to say, unfortunately, that the jury is still out on a number of different issues here, and so the relationship between different groups of arthropods is still quite contentious. Here's the old view here. The old view was that there was a group of arthropods that had two branched or biramous appendages here, okay, the trilobites, the crustaceans, the glycerids, and there's a group of another separate branch of uniramous animals, animals with just one branch to their appendages, such as the insects. So the uniramia or the biramia were two separate groups, okay? That's an old view, that is outdated. People no longer believe that. In part, what happens now is that the branching tree is thought to involve the insects, the uniramians, and the crustaceans together being basically on the same part of the tree here because they both possess mandibles. Hence, they'd be united into a group called the mandibulates. Okay, and here's the crustaceans with their unique features, Nauplius larvae, right? Two pairs of antennae, gnatho-based mandibles, and so on. And here's the uniramians with their whole limb mandibles, Malpighian tubules, etc. Now, even this, however, is outdated. Current evidence suggests that insects actually arose here within the crustacean groups, they're really just terrestrial crustaceans. Okay, that's what the most recent molecular evidence suggests. In terms of insects, there's an enormous number of insects on our planet. They are really the dominant animals out there. And unfortunately, we just don't have enough time really to do this justice. So all of these amazing groups of animals, the collembolans, the thysanurans, the odonatans, and so on, they all will be best studied by you by taking a whole other course called entomology. And I encourage you to do that if you're interested in insects. Finally, I want to stop on an interesting developmental note. It turns out that development of insects, and especially insect limbs or legs, where those legs are located, are controlled by these master genes, these homeobox genes. And mutations in these genes might literally lead to a very strange feature, okay? A pair of legs that is found in a very unusual spot. Let's take, for example, the common housefly, Drosophila, right? Here's the head end here of a normal-looking wild type, we call it, Drosophila here. And you can see here where the antennae are located, which are very small in the head. But in the Antennapedia mutant, what we have here is legs, pedia, in place of the antennae, right here, sticking way off the head. It's pretty fantastical. And what does this mostly tell us? Mostly it tells us something about how these master genes are controlling which legs go on which segments. And as we've talked about quite a bit with our arthropod lectures, different arthropods are all about that. They're all about which legs are on which segments, whether they're chelate or clawed or not, whether they're biramous or not, and so on. Okay, I'll stop there, and we'll move on next to talk about a new group of animals, the Lophophorids.